Brant Isakson is here now, the 2017 APS Bowditch Award Lecturer. First of all, congratulations on the honor.
Thank you very much.
So tell us a little bit about your talk.
Yeah, so today I'm going to be talking about one of the themes that has been throughout my graduate career, through my postdoc, and my time as a faculty member, and that's how two different cell types talk to each other, and in particular in the blood vessel wall. And one of the things that we've really had fun with is trying to figure out how this may change in different states, disease states, and in particular how we can take advantage of this at the translational level, so how we can maybe design some new pharmacological targets to treat drugs. In this case, I'm going to be talking about how we could alter hypertension. So, yeah.
Which is tremendous and impacts so many people in this country.
It's a start. I think we have something new, something interesting, and it'll be exciting to kind of give this talk and get some feedback and see what people think.
The basic concept you have been so passionate about for so long, but it seems like it has so many other implications. As people listen to you talk about this research, what do you hope they take away from it?
Well, I think broadly, heterocellular communication is really something that all cells have to be able to do, and they have to coordinate correctly. You can see that when two different cell types don't talk together correctly, you have a lot of things that don't go right, and so you have like problems with cell proliferation. So pulmonary hypertension is a great example of that. People have looked at how changes in heterocellular communication can cause smooth muscle cell proliferation, which can have deleterious effects. Other examples, of course, cancer. If cells don't talk to each other correctly, the cells can proliferate in not a good way. So I think that these are all extreme examples, of course, but every cell type has to be able to talk with other cell types correctly, and it's a very coordinated, it's a very beautiful thing how they're able to very specifically talk to each other using certain molecules. So yeah, and hopefully part of my lecture that comes across is I'm very focused on one particular system in the vasculature, but I hope that people can kind of see that throughout whatever system they're working on. That could certainly apply to what they're studying.
And you think that's why it's so important for scientists to come to meetings like this, to sort of inspire other people to go beyond?
Certainly. I think one of the great things about this meeting is that you pick up on ideas that you may have floated around in your head, or you may have caught a glimpse of that, but here you can really kind of flush it out a little bit. You can go to a wide range of lectures that are kind of outside of your wheelhouse, but that may apply to some ideas that you've been thinking about. And generally, I think what's also been great for me also is the amount of collaborations that come out of this so quickly. Everyone here is very willing to talk about their work. They want to talk about their work. And so it creates one of these classic scientific environments where people want to share. People want to understand that the whole goal is to do the best science. Everyone is really excited about the work. So,
from your experience, going beyond the excitement and the knowledge that you gain from being here, how does it then ultimately impact your career after being here?
That's great. You know, I've been coming to this meeting since 2001, when I was a PhD student, and I've come every year ever since. And it's just a meeting that's on my calendar. I don't miss it. And what's really great is I come to this meeting, and there's people who I only see maybe once a year, but it's at this meeting. And so it's a time for us to reconnect, catch up quickly. What's new in the field? What are you doing new? You know, what am I doing new? And then kind of share some ideas. And you know, it's a great time to kind of keep building that scientific rapport. And I think that as you stay with the meeting, as you keep coming to the meeting, you start meeting more and more and more people, and you can build these collaborations, really build into something very meaningful. So you can take someone who works on one particular protein and then someone who works on another, and the two of you together can do something much better than individually. So, yeah.
What do you think is the key to making the most out of it while you're here?
I think the key is to really move around, and that is, you know, the amount of sessions that they have here, the amount of talks. It's a cross-society meeting, so you can go to a plethora of talks going on at any one time. see how they fit into your research, venture into something new, go to a talk by ASPED, by APS, by any one of these organizations. Get out of your wheelhouse a little bit, but see what they're thinking, how they're thinking about it, and then how that applies to you. And of course, besides that, of course, talk with people. The poster sessions, go talk with people at the posters. This is ultimately about making contacts, and so that's what I would recommend.
And advancing science. Thank
That's
you.
right.
Great advice.
Thank you.